Hi everyone, we are the group 5 and we're gonna talk about the sterilization. And for the distribution of the reports and members, I let Trisha and Doc will talk about the physical method of sterilization, and Erica or Pilia for chemical method, and Erica Santiago is for the procedure, and the last is the result for Vanessa Tumaneng. So let's discuss first what is the sterilization. So sterilization is the condition of being sterilized, the destruction of microorganisms as pathogenic or saprophytic bacteria, vegetative forms and spores. And now, Isla will start her report.
Let's go on to physical method. So, heating is the most common physical method of sterilization. Number 1, we have the moist heat. It is preferred over dry heat because of its more rapid killing action. And it is classified according to the temperature used. Letter A, the temperature below 100 degrees Celsius. The pasteurization is the process where packed and unpacked products such as milks and other beverages are treated mild heat. So, it is used to eliminate pathogens and extend shelf life. Number 2, the conventional method. It is heated at 60 to 65 degrees Celsius followed by rapid cooling. Number 3, flash method. It is heated at 72 degrees Celsius for 15 seconds followed by cooling at 13 degrees Celsius. And the newer pasteurization was developed and it is called ultra high temperature or UHT. It is heated at 140 degrees Celsius for 15 seconds and 149 degrees Celsius for 100 degrees Celsius. We have the boiling and the fractional sterilization or tinderization. In boiling, it is uses water at boiling point of 100 degrees Celsius. And syempre, alam naman natin to, boiling, di ba? And then the fractional sterilization or tinderization, it is the intermittent sterilization and involves exposing the material. And this fractional can be used as sterilization of culture media such as above 100 degrees Celsius, autoclave or steam under pressure. It is the most efficient method of sterilization because it can destroy all microbial forms. The temperature for sterilizing is dependent on the pressure of the steam. So, because of its higher temperature and pressure, it would only take 15 to 20 minutes after sterilizing. Let's go to dry heat. So, it is the effectiveness depends on the penetration of it through the materials sterilized. So, it is used to sterilize materials in enclosed tubes such as test tubes and petri dishes. So, dry heat contains open flame, incineration, and lastly, the infrared rays.
Chemical methods. Chemicals can inhibit the growth of pathogenic organisms either temporarily or permanently. There are several factors that can affect the efficiency of chemical agent. First, concentration and potency of the chemical agent. Second, duration of exposure. Third, temperature. Fourth, nature of the surrounding medium. 5. Nature of the organism. 6. Nature of organisms. Size of inoculum. Under chemical method, there is surface active agents. These used as disinfectants in homes and hospitals, but their activity is reduced in the presence of hard water and organic matter. Under this agent, there is cationic agent and anionic agent. Cationic agent is quaternary ammonium compound, while anionic agents are soaps. Phenolic compound disrupt membranes and denature proteins. Under phenolic compound, there is phenol, cresol, chlorhexidine. Phenol are bactericidal and fungicidal. Cresol are phenol derivatives safer than phenol. Chlorhexidine is antiseptic hand wash. Alcohols dehydrate cell, disrupt membranes, and cause coagulation of protein. Under alcohols, there is ethyl alcohol used as skin antiseptic, isopropyl alcohol, it kills bacteria, benzyl alcohol used for preservation, methyl alcohol, it kills fungi and spores, used in disinfecting, inoculating foods. Denaturing agents includes acids and alkalis, alcohol and acetone, phenol and cresol. Modification of the functional group of protein and nucleic acid. Heavy metals are mostly bacteriostatic than bactericidal. Under heavy metals, there is mercurial used as antiseptic, and silver compound are bactericidal. Second, halogens. Halogens are bactericidal, oxidizing agent. Under halogens, there is chlorine. Chlorine used for water disinfection, iodine used as antiseptic, hydrogen peroxide used for cleaning wounds. Third, alkylating agent. Aldehydes. It kills all microorganisms including spores. Formaldehydes or formalin used to sterile beddings and furnitures. Glutaraldehyde, it kills spores and used in sterilizing medical equipment. Ethylene oxide, it is also a sporicidal, or it kills spores.
Number 1. Treat the 4 culture tube. Transfer loopful suspension of microorganisms from each tube to another tube of nutrient broth and incubate at 35 degrees Celsius for 24 hours. Record growth with subculture tubes. No growth, minimal growth, moderate growth, or heavy growth. Divide and label nutrient agar play into 4 quadrants and the name of the disinfectant to be used. Transfer loopful of SA rows and label each test tube. Stand 3 minutes after addition of chemical agents. After 3 minutes, transfer loopful suspension from control test tube and strike on quadrant number 1. Get another loopful and strike on quadrant number 2. Get another loopful and strike on number 3. Finally, get another loopful and strike on number 4. Remember to sterilize wire loops in between each step to avoid contamination. Incubate at 35 degrees Celsius for 24 hours. Observe and record growth result. Positive, negative with growth.
Topic, let us all know the result of the video that we've watched last meeting. Let us all know if Bacillus subtilis and Escherichia coli obtain growth when we expose them to heat by the autoclave with 15 psi for 5 minutes, by the boiling water for 30 minutes, by heating with 60 degree for 1 hour. When we are going to expose both Bacillus subtilis and Escherichia coli to the autoclave with 15 psi for 5 minutes, It will not obtain growth because when the pressure reaches 15 psi, the temperature inside the vessel reaches 121 degrees Celsius because of its high temperature. It would take 15 to 20 minutes to sterile the material. It only says that 5 minutes of heating through the autoclave is not enough to obtain growth. The same with the boiling water and heating. It does not obtain growth because boiling water contains 100 degrees Celsius for 15 minutes. It means that both Bacillus subtilis and Escherichia coli is possible to obtain growth with that high degree of heat. About heating at 60 degrees for 1 hour, this method is quite straightforward. It's only good of killing viruses, not for obtaining growth.